Yeah. Good morning. Good morning to all. Good morning to everybody. Okay. So Rita Kumari, good morning. Mindful Soul, good morning. So Pratesh, everyone. So Chalapati Korra, she's you said your name, I forgot. Yeah, good morning to everybody. Yes, so hope you are all very happy after your result, like 10th class results, and soon we are also going to have our offline classes. Yeah, Gujala Charanya got 540 marks. That's great. So good. So don't worry about your marks, 10th class marks. 10th class marks is not at all a criteria for getting a medical seat. In my career, in my 28 years of career, I have seen people who got just 360 or 340 marks in 10th class, but they became doctors. Okay, so it is all not at all related to your board exam marks. Okay then. Now let us go into our today's discussion. Yesterday I started explaining about cell unit of life. There I said various kinds of organelles and different other details. Yeah, I'll see all your marks at the end of our session. Like I'll close this session by 12:45. Sorry, 12:15. After that I'll see your marks. Many of them have started posting your marks. That's great. So thank you and congratulations for everyone who successfully completed their 10th class and achieved best in your 10th class, like best marks in your 10th class. So that is it. That itself indicates like your strong dedication towards your career. So that's great. Keep the same tempo even after coming to our offline classes, which will be started very soon. So come back now. Now you can switch off chat box. Switch off the chat box. Yeah. Now your chat box has been closed. Now listen to me carefully. Kiranya, yeah. Now listen to me carefully. Yes. Now I think yesterday just I told you about different types of cell organelles present in the living cells, including amembranous organelles, single membrane bounded organelles and double membrane bounded organelles. Today I am going to give you some basic details of bacterial cell structure, prokaryotic cell structure. Listen to me carefully. This is very interesting and important also. So you will get many details here. So this is all very important for your offline classes, because once you come for offline classes, we'll be discussing about biological classification, which is a chapter which is actually second chapter in our NCERT +1 book. So in that you are going to have more details about this bacteria, but there they did not mention about the cell structure. So most of the students, they feel difficult in understanding Monera, which is related to the bacteria. I mean bacterial prokaryotes. So most of the I am observing like every year, like most of the students feel tough understanding this Monera concept because they are not aware about the bacterial cell structure. That's why today I am decided to explain about the bacterial cell structure so that you will be more comfortable in your offline classes when the teacher discuss about bacteria, I mean to say Monera kingdom under biological classification. Now listen to me here. So bacterial cells like prokaryotic cells, I am particularly saying about prokaryotic cells, except mycoplasma, all other prokaryotes have cell wall. Remember, Only mycoplasma lack a cell wall. They don't have cell wall. Understood? Except mycoplasma, remaining all prokaryotes have a cell wall. So, if you take a bacterial cell structure I am discussing now. So, a bacterial cell, it has a cell wall. This is cell wall of bacteria. And I already told when I discussed about Monera, like 5 kingdom system of classification, bacterial cell wall is composed of protein, amino acids and polysaccharides. Simply we can say that it is made up of peptidoglycan. What? Peptidoglycan. It is non-cellulosic and it is peptidoglycan. That is the cell wall. Cell wall has some functions, so we will discuss later about the functions of cell wall. Simply to say, it provide definite shape to the cell, also protect the cells from bursting as well as collapsing. Understood? Several other functions are also there. So I am not going to that details, just I am giving you what are the structures present in a bacterial cell. Now, below this cell wall, there is plasma membrane. Now look at here, this is plasma membrane. This is plasma membrane. So I said this is cell wall. I said the cell wall is non-cellulosic and it is composed of peptidoglycan. In most, there is some difference also there in different bacteria and different groups of bacteria in cell wall composition. Most common is peptidoglycan. Next, below to the cell wall, there is a cell membrane, or you can also call it as plasma membrane. Now, the most important thing, the cell membrane is structurally very much similar to, functionally also, very much similar to eukaryotic cell membrane. Eukaryotic cell membrane. So, it is very much similar. So, we can say it is chemically lipoproteinaceous. That means it is composed of, it is made with lipids and proteins. Lipids and proteins. So that is the importance of this plasma membrane. That is why it is called lipoproteinaceous membrane. Understood? So it is a lipoproteinaceous membrane. Now, so this cell membrane is selectively permeable in nature. What nature is there? Selectively permeable. It is selectively permeable, which means it allows only a selected molecules to pass through from one side to other side, like the molecule present outside to inside or inside to outside. Only selected molecules will be allowed by the plasma membrane. What are that molecules, how it sends, this are all different story. So these details you can know when we discuss about plasma membrane structure. Just remember that it is a selectively permeable membrane. Okay, I am not going in detail of every word. There is a lot of information about each and every word now I am discussing here, but just try to remember only the terminology that I am using here. Don't go in detail for this kind of terminology. Now, apart from this plasma membrane, outer to the plasma membrane, just outside to the plasma membrane, in some bacteria, not in all, in some bacteria, there is another layer which is called glycocalyx. This is glycocalyx. This glycocalyx again exists in two different forms. It may be present in the form of slime layer, or it may be present in the form of capsule. In any one form it will be there. No bacteria possess both slime layer and capsule. A bacteria will contain either slime layer or capsule. There are also bacteria without this glycocalyx, remember. There are some bacteria which don't have glycocalyx. Some bacteria have glycocalyx. If glycocalyx is present, then it may be present in the form of slime layer. It may be present in the form of slime layer, or it may be present in the form of capsule. What is this slime layer? Slime layer in the sense, like if this glycocalyx is found in the form of a loose sheet. How? Loose sheet, then it is called slime layer. Capsule, if it is present in the form of a tough or rigid layer, then it is called capsule. What is this glycocalyx? Glycocalyx is also a part of envelope. It is composed of polysaccharides and proteins. With what it is made? Polysaccharides and proteins. And remember, these three layers, what are they? This glycocalyx, cell wall and plasma membrane, together called cell envelope. What is it called? Cell envelope. So, how many cell envelopes are present for a bacterial cell? How many cell envelopes? Is it 3 or 1? 1. There is one cell envelope. This one cell envelope has 3 layers. Have you got the clarity? One cell envelope containing 3 layers. These 3 layers together called envelope. Got it? So that is the importance of this. Is it clear for all of you? Yeah. Now inside the plasma membrane, like inside the cell, there is a semi-fluid matrix. I already told in the last class. This semi-fluid matrix is also called cytoplasm. So cytoplasm is a semi-fluid matrix. Okay. Then in this cytoplasm, and you know that prokaryotes, what do you mean by pro here? Primitive. Karyon means nucleus. That means they have what kind of nucleus? Primitive nucleus. What do you mean by primitive nucleus here? Their nucleus is not surrounded by any envelope. That means nuclear envelope is absent. Or simply to say, here the genetic material is a double-stranded DNA. What is it? Double-stranded DNA. This DNA or genetic material, you can also call it as chromosomal DNA, which is present directly in the cytoplasm. Look at here. Now, this is genomic DNA. What is this? Genomic DNA or chromosomal DNA. Chromosomal DNA. This chromosomal DNA is present directly in the cytoplasm. This cytoplasmic region Listen to me carefully. This cytoplasmic region containing this chromosomal DNA is called nucleoid. What is it called? Nucleoid. So what is nucleoid? Cytoplasmic region containing chromosomal DNA. The region of cytoplasm containing genomic DNA is called nucleoid. And if you observe this diagram, in the plasma membrane I have shown some inward folding. Can you see here? Inward folding. Yes, this is very important here. In some bacteria, in some cases, not in all, in some bacteria, this plasma membrane shows an inward folding. What is it? Inward folding. Now this inward folding itself is called mesosome. Mesosome. So mesosome is an inward folding of the plasma membrane. Is it clear? Mesosome is an inward folding. It has many functions. Or you can also say it is a differentiated form of the plasma membrane. Differentiated form of the plasma membrane, or it is an inward folding of the plasma membrane, or it is a specialized form of the plasma membrane. It has many functions. Of course, I am not going into details of functions. Just remember that plasma membrane is also folded inwardly, like towards the cytoplasm. This folding itself, this folded plasma membrane itself is called mesosome. Is it clear for all of you? Yeah. Now, I said there is a chromosomal DNA. How many chromosomal DNAs are present? Only one. So it is single and it is also double stranded. Remember, double stranded. You can also simply represent this double stranded as DS. DS. Double stranded means there are two strands with two threads. Like you can see the diagram. This is one thread, this is another thread. These two threads are folded like this. So it is double stranded circular DNA. Circular, naked in the sense it is not covered by any nuclear envelope. So naked DNA. This is genomic or chromosomal DNA. And remember, many bacteria, not all, many bacteria, in addition to this chromosomal DNA, they contain some small, look at here, small, circular, double-stranded, small, circular, double-stranded, naked, extrachromosomal, self-replicatable, DNAs called plasmid DNA. Remember, so how many types of DNAs are present in the bacterial cell? How many types of DNAs are present? Two types. What are they? Chromosomal DNA, plasmid DNA. Which one is larger? Chromosomal DNA or genomic DNA. Which is smaller? Plasmid DNA. Both are circular. Circular means they are not linear, they are not straight. The two ends are joined here. Both are circular, both are naked, and both have self-replication, but this plasmid DNA is present outside of the chromosomal DNA. It is not a part of chromosomal DNA. Remember, that is why it is called extra chromosomal DNA. Extra in the sense outside, outside the chromosomal DNA. So it is extra chromosomal DNA, self-replicatable DNA, that is called plasmid DNA. Plasmid DNA is also having several important functions. It confers so many important functions to the bacterial cell. I'll give you one best example, that it provides antibiotic resistance to the bacteria. I think you're all one day or other day, like in your course of your life, you might have exposed to some bacterial infection. Say for example, you got some throat infection or cough, something like, and doctor may have suggested you to take azithromycin, right, or some other antibiotic. So you took that, after some time you get the relief. Again, if you expose to the same kind of infection after some time, if you take the azithromycin, same antibiotic, again it may be suppressed, but not too fastly. It takes time. If you go for third time again the same antibiotic, then this antibiotic may not works for you. Why? Because this bacteria start acquiring resistance against to this azithromycin. Then if you go to the doctor, he has to suggest another antibiotic for you. That means this antibiotic resistance is present in plasmid DNA. Understood? So this resistance is acquired by the bacteria. In the course of time, as they expose to a particular chemical, it gradually acquires. How it acquires and what is the function of this antibiotic resistance, this is all discussed under biotechnology chapter that will come in your second year, like +2 syllabus. Now come back. So this is what you can see with regard to the plasmid DNA. In addition to this, you can also find 70S ribosomes. What kind of ribosomes? 70S ribosomes. Now ribosome is the only organelle which is present in the bacteria. This ribosome, I already told in the last class that it is not a membrane bounded organelle. It is a membranous organelle, and it is 70S. Here S represents its sedimentation coefficient, or simply to say, it is an inverse measure for its density and mass. Its density and mass. Okay, so that is the importance of this 70S ribosome. So it is a measurement for its density and mass. It reflects its density and mass, not inverse. I am sorry. So it reflects about its density and mass. So that is the importance of 70S ribosome. Don't worry about the 70S and 80S. I will give you the detailed explanation about the 70S and 80S. Yesterday also I told different types of ribosomes. These details we will discuss in our offline classes, like what exactly this S represents. So S in the sense here, Swedberg unit. Swedberg unit is a kind of unit used to measure its density and mass. So how it is estimated and why we are using this S, and what exactly denotes. So these details we are going to discuss in our offline classes when we discuss about ribosomes. So just remember, they have only 70S ribosomes. Which one? Bacteria. So this is another important thing we have to know. Next, apart from this, you can also find bacterial cells also contain inclusion bodies. Inclusion bodies in the sense reserved food materials. So this is an inclusion body. Inclusion body. So it is a reserved material. Reserved materials. Reserved materials in the sense it may be glycogen or sometimes phosphate granules. Understood? Sometimes cyanophycean granules. There are different types of inclusion bodies are also present. Apart from this, they also have some, if bacteria is motile, motile means locomotory, then the bacteria possess some locomotory structures. What are they? They are nothing but flagella. Now this is the flagella. So this is flagella. So flagella helps in locomotion. So it is a filamentous extension of cell wall, and this flagella has three important parts. One is basal body, and other one is hook, and other one is filament. See, here you can find the basal body. This is basal body, and this is a hook. Yeah, let me represent this. This is basal body, and this is hook. This is hook, and this is filament. Filament. These are the three structures: basal body, and this is hook, and this is filament. And it helps in locomotion. There is a great variation with respect to its distribution and number also. Apart from this, bacteria also possess pili or pilus. Look at here. This is pilus or pili. Pili is plural. Pilus is singular. Pilus is singular. This pilus is an elongated tubule made with a special protein called pillin. This pilus helps in sexual reproduction of bacteria. Bacteria also have sexual reproduction. A method of sexual reproduction existed in bacteria is conjugation. Conjugation is a method of sexual reproduction. In that, this pilus helps. How it helps, what exactly its function, like its structural details, this all we'll discuss later. Just remember that they also have pilus, and pilus helps in conjugation method of sexual reproduction. Then, apart from this pilus, you can also find in some bacteria there is a bristle-like fibers. Bristle-like fibers. Now these fibers are called fimbriae. This fimbriae helps in attachment of bacteria. So attachment of bacteria to the surface of rock or to a host tissue. Okay. So this is about fimbriae. These are the most important things that we need to discuss. We need to understand about bacterial cell structure. See, this bacterial cell structure itself takes almost 3 hours to complete. 3 class hours. So I just gave only basic details without touching the complete details of this bacterial cell structure. Just I gave only the terminology associated with the structural details of bacteria. Okay, so we'll discuss in detail once you come for the offline, but try to remember this terminology whatever I have discussed today, and just copy this diagram into your notebook. Like you can take first a screenshot and copy it into your notebook so that it will be helpful when you come for offline and when teacher starts discussing about Monera under biological classification chapter. Okay, so just copy it, just take the screenshot and copy it, and copy it into your notebook. Now you can ask your doubts. If you have any doubts about today's topic or yesterday's topic, you can now ask me. Your chat box is opened. Yeah, is it completed? Now you can ask. Sujata, no doubt. Thank you. That's great. It seems you are following the classes very carefully. Yeah. Srikant Yadav, what do you want? Inclusion bodies is also a kind of cell. No, except ribosomes. Ribosome is the only, Karthik, listen to me carefully. Ribosome is the only organelle present in the bacterial cell. No other organelles are present. Okay. One guy is asking me, Junior Itachi, why, who told you that tomorrow will be the last day for your online classes? Did I tell you? No. So it may continue till June first week. Don't worry. Now listen to me here. So Karthik is asking me about inclusion bodies is organelle. No. Bacteria or prokaryotes have only one organelle, that is 70S ribosome. Inclusion body is a reserved food. Reserved food or reserved substance. Reserved means stored. Stored food, a stored substance, like sometimes phosphate may be stored. Understood? Phosphate may be stored. Sometimes glycogen may be stored. By glycogen may be stored in the form of polybeta hydroxybutyrate. So that stored substance itself is called inclusion body. Is it an organelle? No. Can you call glucose that you are eating every day or carbohydrate that you are eating every day as a breakfast, lunch and dinner, can you call it as an organelle? No. Mr. Karthik, did you got my explanation? Yeah. Zoology ma'am said, oh, I don't know whether she might be knowing information from the academicians, but I am not aware about this information. I think so, the classes will continue till June first week, because your offline classes will start maybe in the June first week. So until that may be there. I am not aware about it. So if there is no classes, then your administrators will inform you. Okay, so don't ask about about these classes, online classes, whether they continued or ended. Don't ask me about that. I am not aware about it. I told many times that I am not involving in any administrative issues, and I am purely a teacher. Okay, so except teaching, except my subject, I am not aware about anything. Next. Sir, inclusion body is like a vacuole? No. It is not a vacuole. Bacteria, some bacteria have gas vacuoles. Gas vacuole may be there, but this gas vacuole is not like a normal vacuole that you see in case of plant cells. So this gas vacuole, they are filled with gases. So that is different. And gas vacuole is also not a organelle. It is also not a organelle. Pili and Fimbria is same. No, Rita Kumari, it's a good question. Pili and Fimbria, entirely different. Pili is different, Fimbria is different, but only one function is same, that both are meant for attachment. Pili helps in attachment of two different bacteria. Fimbriae helps in attachment of a bacteria to the surface of host or to the surface of rocks. Is it clear? Rita Kumari, is it clear for you? Yeah. Sir, can you explain again about plasmid DNA? Yes. Kurra Anuradha. Yes. Listen to me. Plasmid DNA is present in many bacteria, not in all, many bacteria. They are small bacteria, small DNA. They are not the part of this genomic DNA. They are present outside this genomic DNA. They can replicate by their own. Understood? They also have some genes to regulate some of the activities of the cell. I told you one example that these plasmids may contain resistant genes for certain antibiotics. Resistant genes for certain antibiotics. That is the importance of this plasmid. So, plasmid DNA and genomic DNA both can replicate, both are double stranded, both are circular, both are naked. But plasmid is not a main DNA. It's a small DNA, have only some functions. Whereas genomic DNA is the main DNA that controls almost all the functions of the cells. Almost all, not all. Okay. Some functions will be under the control of plasmid DNA. So that is about plasmid DNA and genomic DNA. Is it clear for you, Miss Anuradha? Yeah. Sir, in which cell organelle will make harm to other animals? Which cell organelle will make harm to other? Okay. So you are asking about organelle that can harm other animals. Oh. It is not like there is no specific organelle that can harm unrelated animal. But there is one organelle in eukaryotes which is called a lysosome. Listen to me. Who is that? Question asked. Junior Itachi. Why don't you keep your own names? Yeah. So, Mr. or Miss, I don't know you. So, there is one organelle in eukaryotes which is lysosome. Lysosome can cause digestion of other organelle when cell become old or non-functional. If, say for example, there are some once the cell become old and some mitochondria also become, when mitochondria become old and become non-functional, this non-functional and older mitochondria are digested by lysosomes. Or if the cell under starvation condition, this lysosomal membrane ruptures and it releases all the enzymes into the cell and digests the whole cell, so that the cell will be killed. This process is called autolysis and it is carried by lysosomes. That's why lysosomes are called cell suicidal bags. Okay. So don't worry, we'll discuss all these details in our offline classes. I'll be going very slowly in offline classes. Okay. I will inject each and every word of NCERT book in detail. So once you come for the offline classes, you can see and feel the taste of botany. Okay. So come back to the next one. The next question, Mr. or Miss Hangama. If the classes continues, then how we can prepare about for our second phase preparation of CBSE? Yes, that may be one of the reason for CBSE people. I think they have their mathematics exam soon. I think this month, next week or some, so I don't know exactly, mostly next week. That may be one of the reason. Okay, I am not aware, and this detail like whether classes are continued or not continued, this is all explained by your administrators. Just ask them, they will tell. Chalapati Korra, oh that's good. Kiranya, you got 555 out of 600. That's great. Congratulations, and that's really a great achievement. Okay, God bless you. Next, yeah, that's what Mr. Hangama or Miss Hangama, I don't know. So your gender, you ask to the administrators, they will inform you about exact date of ending this online classes. Okay. Next, any more? Sujata, you got 547. That's great. Congratulations. Okay. To whom will teach you girls campus or boys campus? Okay. You tell me, where should I come? Should I come to you or shall I go to boys campus? I think so you are girl. If you are girl, yes, Karthik, you teach girls in campus or boys campus. Okay. You decide and tell me to whom I should come. Okay. So you decide, don't tell me, tell to your administrators. They will take the decision. So the ball is not in my court. Okay. So in their court and your court. So leave about this matter. So some saying boys, some saying girls, that is different. Okay. So how the sections are divided? Why are you worrying about sections, madam? Sujata, don't worry about sections. Okay. So whatever may be the section you are, wherever maybe you are, you have to do your work and you have to, dedicate completely on your studies. And we are there to guide you, to enrich your knowledge and to get a medical seat. Don't worry. Next. Okay. So you are Guy, Junior Itachi. So you are asking me to come boys campus? Oh, that's great. Okay then. Any more doubts? Any more doubts? Leave about this unnecessary discussion. Okay. So just come to our today's topic. Jagriti. Oh, Jagriti, it's extraordinary. You got 588. That's great. Don't worry about your 10th class marks, whether you got 547 or 600. So that is not at all a question. Okay. So don't worry about your marks. Just keep your concentration for your future plans, like for your next endeavor that you are going to face. What is that? Getting a medical seat. Keep your concentration only on getting medical seat. Okay, thank you very much. And Nagi Reddy has got 585. That's great. That's great, really great. Yeah. So Lakshman 566. So I think it seems this time Gosala campus. Filled with only the meritorious students of total Andhra Pradesh and Tamil Nadu as well as the total India. So many of them have got more than 500. That's really impressive and a great achievement. So God bless you. Congratulations once again to all of you for your great achievement in your 10th class board exams, and at the same time all the best for your future endeavors. Still you have to, just only one phase is completed. There is second, third phase is also there. So still you need to do lot of work. Okay. So God bless you. So with this I am closing today's session, and tomorrow we'll meet again at the same time. Thank you all. Bye.